Good morning, it's December 14th, and this is your daily brief in science. Here's everything you need to know. Scientists have conducted extensive studies on the mouse brain to understand its epigenomic and transcriptomic landscape. Regulatory elements that shape gene regulation and epigenetic landscapes were discovered, with a focus on transposable elements and transcription factors. User-friendly platforms for data visualization and exploration have been developed, aiding comprehension of neuronal organization and diversity. The studies emphasize the need for standardized data collection and shared data standards for global projects. Powerful atlases mapping the composition of the mouse and human brain have been created, offering insights into brain evolution, basic biology, and potential treatments for neurological disorders. The Human Cell Atlas project aims to create a comprehensive map of the human body, representing full cellular diversity. These atlases are seen as the beginning of a scientific revolution in understanding cells and their functions. A recent study has identified the hormone GDF15 as the primary cause of severe morning sickness, or Hyperemesis gravidarum, in pregnant women. Women with hyperemesis had significantly higher levels of GDF15 during pregnancy. The hormone's effects are dependent on a woman's sensitivity and prior exposure to GDF15. The study provides hope for better treatments and prevention strategies for Hyperemesis gravidarum, including potential medications to block the hormone's effects. Genetic factors associated with hyperemesis were also identified, providing further understanding of the condition. The severity of morning sickness is correlated with the amount of GDF15 produced by the placenta and the woman's sensitivity to the hormone. Current treatments for morning sickness include anti-sickness medicines and steroids, and in severe cases, hospitalization and intravenous fluids may be necessary. Researchers are seeking pharmaceutical partners to further explore potential treatment and prevention options. NASA engineers are addressing a computer glitch on Voyager 1 spacecraft. The glitch affects the flight data system and telecommunications unit, resulting in repetitive data transmission. Efforts to restart the system have been unsuccessful, requiring several weeks for a new plan. Despite the glitch, Voyager 1 continues to provide valuable data about interstellar space and the sun's protective bubble. At 15 billion miles away from Earth, Voyager 1 is the longest operating and farthest traveled mission. Technicians are resorting to decades-old manuals in their search for a solution. A recent study in Nature used whole genome long-read sequencing to examine genomic structural variation in Indigenous Australians. The study found a high number of unique structural variants, primarily consisting of repetitive sequences specific to Indigenous Australians and individual communities. Research identified novel repeat expansion sites within protein-coding genes and potential functional variants related to type 2 diabetes in Indigenous Australian populations. The study emphasized the need for more inclusive genomic studies for equitable healthcare and the importance of capturing the unique genetic variation of Indigenous Australians in a comprehensive database. The research highlighted the importance of involving Indigenous Australian peoples in genomic research and respecting cultural perspectives. Sharing genomic data through a community consultation framework can support efforts to enhance genetic representation and healthcare equity. Understanding the genetic differences between Indigenous communities can provide insights into their linguistic diversity and their past. A new study found that individuals with diabetes are more susceptible to respiratory viral infections due to high blood sugar levels. Hyperglycemia impairs the function of lung dendritic cells, leading to a defective immune response, delayed viral clearance, and increased mortality. A study on mice showed that high blood sugar levels resulted in higher mortality rates and impaired immune responses. Insulin treatment to normalize blood glucose levels improved viral clearance and immune response. Another research found that high blood sugar levels disrupt the function of immune cells in the lungs, increasing susceptibility to severe lung disease when infected with viruses like COVID-19. Tight control of blood sugar levels, or the administration of small molecules, could restore immune cell function and reduce the risk of severe lung infection. These findings have significant clinical implications, especially considering the large number of people worldwide affected by diabetes. Further human clinical testing is needed to explore the potential of these treatments. This has been your Daily Brief in Science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates at www.brief.news. And for more daily podcasts about the topics you love, visit www.brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.